Hello everyone, my name is Madhura Nayak and I will be your presenter today. Today's webinar focuses on introducing the RNAscope 2.5 HD Duplex Assay. This webinar will be a joint presentation with our in-house R&D scientist. A special thanks to Dr. Ming-Xiao Hu for joining us today and presenting in this webinar series. A few key points to note. You will be muted during this presentation. Please use the chat feature available to you to ask your questions while the presentation is in progress. If we cannot answer your question during this webinar, we will get back to you offline. We will begin this webinar by reviewing the key basics of RNAscope technology. We will then introduce you to our new RNAscope Duplex Assay and review the product configuration and the technical details. The next section focuses on understanding a few of the many applications this assay can be utilized for your research interests. And then Ming-Xiao will delve into the data review performed by our R&D team. Let's begin with the overview of the technology. RNAscope is based on proprietary in situ hybridization technology based on unique probe design and signal amplification. This platform has the sensitivity to detect most genes in situ as well as simultaneously quantify multiple RNA transcripts. Just as any in situ assay, the RNAscope assay involves permeabilizing properly prepared samples to allow complementary oligo probes to bind to RNA target of interest. Specific signal is amplified using the double Z probe system, which is subsequently detected and quantified. The assay is extremely universal as well as rapid and scalable. To design specific probe for your target of interest, ACD requests you to provide us either the accession number information or the exact sequence of the target. You could even indicate your region of interest for targeting with start and or stop positions. Our bioinformatics scientists channel this information through the proprietary ACD alignment algorithm to select optimal sequences to ensure target specificity and uniformity of oligohybridization in the assay. The algorithm evaluates the melting temperature of each oligo in the pool to ensure no cross-hybridization with non-target RNA under stringent RNAscope conditions. So how does the technology work? RNAscope probe design is the key. We use a double Z probe design. There are three components of the Z. The upper region is the 14 base tail sequence, and that is the region where pre-amplifier and the cascade of amplifications will occur. The spacer region is in between the base and the other end, allows the flexibility for probe binding efficiency to mRNA. The lower region of the Z binds directly to mRNA. This lower region contains 8 to 25 base sequence that is specific to your target. Zs are used in pairs. In pairs, the upper region of the Zs will provide 28 bases for the preamplifier binding. Both Zs bind adjacent to each other to your target region from the lower region. The yellow line here represents your RNA. If your RNA is intact under ideal permeabilization conditions, the assay will hybridize up to 20 Z probe pairs to your RNA. But for many samples, such as cancer FFPE samples, your RNA could be degraded and fragmented. The simplicity of this design and relatively short probes binding to the target allows for binding to even short pieces of RNA. What our scientists have demonstrated in a lot of experiments is that we just need 3 pairs of Z to bind to the target in order to detect the signal. Even for fragmented or degraded RNA, which are not likely to have migrated and are localized, you should see a signal amplification for a single molecule. And again, this is under ideal conditions. How does the signal amplification work? You have your target transcript. The probes are added. The double Z's land adjacent to each other. The preamplifier will then bind to the top region of 28 bases. Each amplifier will then serially have 20 amplifiers bind to it. On each amplifier, there are 20 labeled probes, 20 times. So we see 20 Z pairs with 20 amplifiers and 20 labels, which is about 8,000 labels per transcript under ideal conditions for a 1 kb region. Now, what about nonspecific binding? If a single Z binds to nonspecific RNA, the preamplifiers cannot be stable on just 14 bases of the upper region. So the nonspecific binding that may occur does not get amplified. And this is how the double Z design prevents background noise and contributes to specificity. Based on this technology, RNAscope assays are applicable to a wide variety of research applications. We have over 430 and above publications to date with research focusing in cancer, neuroscience, and signaling pathways, to name a few. With this background knowledge of the RNAscope technology, let's review the technical product configuration of the new 2.5 duplex assay. It is our pleasure to introduce Advanced Cell Diagnostics' latest product release, the RNAscope 2.5 HD Duplex Reagent Kit. This robust kit was developed based on customer demand and replaces the existing RNAscope Duplex Reagent Kit. The Duplex Kit is a green and red combination using fast red and green, and the probes that are compatible with this kit are in channel 1 and channel 2. A few applications of this kit are for study of gene co-regulation, profile gene expression in a specific cell type, which can be identified using a cell-specific marker, and map co-expression of two targets within the same cellular context based on reliable chromogenic detection of two transcripts. Let's review a few important highlights of the RNAscope 2.5 HD Duplex assay. A few key features are that the assay is based on dual 2.5 chemistry, where both channels are amplified to the same level. Red and green is a color combination of choice, and all reagents are ready to use. The one advantage that this kit has is that the pre-treatment reagents are same as the singleplex assay. Now, these features allow you to visualize low-expressing targets in both channels. It has comparable sensitivity to 2.5 singleplex assays. It's easy to use and no additional optimization is required. This kit results in sharper, crisper signal in both channels, which is easier to quantify and produces better publication quality data. With the reagent kit, you will get 4 boxes. Let's review the reagent kit components. You get a box of the target retrieval solution, one of the protease and the hydrogen peroxide reagents, one of the amplification reagents, and one of the wash buffers. You will also require a Hive-Easy oven to perform the assay under optimal conditions. For the probes, the compatible channels are channel 1 and channel 2, with channel 1 being HRP-based and can be visualized in the green, and channel 2 being AP-based and can be visualized in the red. The two channels are relatively comparable in sensitivity. Due to the property of the red color and per pathologist preference, it is recommended to put a lower expressor in the red channel. We also provide positive controls which are pre-mixed. For those of you who are familiar with the duplex positive control probes for the older Duplex assay, please note the change in the configuration of the pre-mixed probes. For the new Duplex assay, the PPIB or the higher expressor is in the channel 1 and will be visualized in the green. The lower expressor, polar 2A, is in channel 2 and will be visualized in the red. This is keeping in mind our recommendation. The duplex negative control remains the same. It has DAP-B, a negative control probe in both the channels. For new users, we now have an intro pack with all the must-have materials bundled into a package. This comes with the control slides, the control probes, the reagent kit, the wash buffer, the hydrophobic pen, and our recommended mounting medium, the vector mount. It is important to keep in mind the use of vector mount mounting medium, which helps preserve the red and green signal intensity. In order to get started with the duplex assay, for new users, we highly recommend the intro pack. Please ensure you review the checklist for the 2.5 duplex. This is available on the support page to confirm all the required reagents are present before starting the assay. We also recommend you review the user manual and the quick guide to familiarize with the workflow prior to starting the experiment. Now let's briefly review the workflow. Start with properly permeabilizing the samples fixed with our recommended condition of 24 hours at room temperature with 10% NBF. Please note this workflow is for FFPE samples or formalin-fixed paraffin-embedded samples. Using our recommended condition of fixation will ensure the RNA quality is well preserved. The typical workflow starts with baking for 1 hour at 60 degrees, followed by D-pair and hydrogen peroxide to block any endogenous peroxidase activity. The step is followed by target retrieval, which breaks the cross-linking caused by formalin fixation and is subsequently followed by protease digestion. As the RNA is now accessible, probes in channel 1 and 2 in the ratio 1:50 are now hybridized for 2 hours at 40 degrees. The channel 2 probe is amplified to yield the red signal, and then the channel 1 probe is amplified to yield the green signal. There are multiple stopping points that allow you to run the assay as per your convenience. You may stop after baking, D-pair, and target retrieval for up to 24 hours. It is advisable to complete these steps and stop after probe hype to split the assay over 2 days. You will need 5x SSE in order to utilize this optional stopping point. And lastly, please pay special attention to the drying and the mounting step, where again we recommend vector mount for the mounting procedure. In order to analyze and quantify your signals, software analysis for the RNAscope 2.5 HD duplex can be performed with HALO software to provide a cell-by-cell quantitative result. Released by INDICA labs, HALO algorithms are designed for the quantification of complex RNAscope expression data. And HALO software provides interactive results with real-time readout, and the software reports individual cell data and maintains an interactive link between cell metrics and cell imagery. Quantitative scoring with the HALO image analysis software can be performed for whole tissue evaluation of RNAscope staining. Each cell is assigned to a bin based on the number of dots per cell. Clusters are divided by the typical probe signal area to calculate a dot number for the cluster. An 8-score calculation is performed in HALO to assign a single score to an entire tissue section based on the average target expression in this tissue. Now let's briefly review the key workflow highlights. This assay has the ability to split over 2 days. It's an easy transition from the singleplex assay as the pre-treatment conditions for the same FFPE samples remain the same. Recommended that the lower expressor is placed in the C2 channel as the red color is visually stronger. Please note both channels are amplified to the same level. An automated quantification of the signal can be performed using HALO software from INDICA labs. As we are now aware of the product configuration and workflow, let's review a few of the many applications you could use the RNAscope 2.5 HD Duplex Assay for. This kit has been extensively tested in-house on 76 samples and across several different tissue types. To name a few, we have tested the human TMA, mouse tumors, mouse tissues such as liver, lung, kidney, and xenograft tissues. On this slide, you can see a few examples of these tissues run with the control probes. Here we list a few of the many potential target applications you could utilize this duplex assay for. For example, colocalizing a cell type marker to a biological marker of interest, as in this example, the NPY neurons or neuropeptide Y neurons are identified and expression of POMC is studied. Here we see an example of detecting multiple targets of interest, and in particular FOXP3 and CD4, to characterize a tumor microenvironment in FFPE lung cancer tissues. Another potential application is to evaluate different gene expression profiles. Combined with the specificity of RNAscope probe design, in this example of patient-derived xenograft, we can clearly identify mouse stromal cells and the human tumor cells by using species-specific PCAM1 probes. And here we see an example of studying ligand and receptor expression in a tumor microenvironment, and in specific, detection of neuregulin ligand and its receptor ERBB3 in breast cancer tissues. Let's look into greater detail at an application to understand immunotherapy. Here, we present the use of RNAscope for the detection of RNA targets in the tumor microenvironment that are involved in tumor immunology and immunotherapy. RNAscope can be used to identify checkpoint markers such as PD-L1 and immune cell markers such as CD8A. In lung cancer tissue, we see a high abundance of CD8A T cell marker in the tumor and stroma colocalizing with PD-L1. In ovarian cancer tissue, CD8A is restricted only in the tumor and stroma, and PD-L1 only in the immune cells and not the tumor cells. With these few key examples of the application of the duplex assay in mind, I request Ming Xiao to take us through some more data presentation. We tested this new RNAscope 2.5 duplex assay on multiple formalin-fixed paraffin-embedded samples, aka FFPE samples. The samples including culture cell pellets as HeLa cells, multiple samples from human tumors, multiple samples from mouse organs. We also tested on human xenograft in mice. So far, we only tested assay on FFPE tissues, and the test in fresh frozen is still in progress. This assay hasn't been validated in culture cells and PBMC. These slides we show the very specific, unique detection of the duplex assays in HeLa FFPE samples. You can see on the figures on your right, no background was observed when slides were hybridized with the negative control probe step B. On the slide to your left, we can using this detect to detect low expressing targets such as TBP in the green channel and poly2A in the red channel. We observe very strong punctate staining in both channels, and the dot size are very balanced. We just reviewed the detection of immune markers in tumor environment. Now I'm going to review a few slides on ligand and receptor detections, detections in neurologies, and detection in human xenograft in mouse. So these slides, we detect the NRG1 and its receptor ERBB3 in multiple tumor samples. And for examples, on the pictures to your right, you will see NRG1 was expressed by tumor cells, not the stroma cells around the tumor region. And the ERBB3 was detected in the marginal regions of the tumor adjacent to the stroma cells. So using the duplex assay, you can define your markers in the unique populations in the tumor microenvironment, and you can define the marker-specific populations at both channels. So next slides, we can see the specific detection of HGF and its receptor CMAT. HGF and CMAT were both expressed by different populations of the cells within the tumor microenvironment. You can see the HGF was detected in the green channels, and the CMAT was in the red channels, and both expressed by different populations of cells adjacent to each other. The next slide is another example of co-staining of receptors and its ligand. We used the 2.5 duplex assay to detect the ligand CD248 and its receptor beta-type PDGF in tumor microenvironment. You can see the co-expression of these two targets within the same cells, as in the pictures of the left, and you can see the cells can be expressed by the different groups of cells like the pictures of the right. We also validated the assay in the study of neurosciences and other organs. So this is the examples of detecting two markers, LGR5 and GLI1, in both mouse brains and mouse colon. You can see the cell-specific expression of both LGR5 and GLI1, and the markers, the cell markers of different cell populations are labeled by the two-channel cell probe very specifically. This slide is reviews the application of the duplex assay in the study of patient-derived xenografts. So in the study of xenografts, it has been challenging to separate targets from different species due to the high homology between each other, such as human and mouse. So in this case, the tumor cells are tumor origin and growing in the microenvironment of the mouse stroma. To separate the expressions of the tumor origin targets and mouse
origin targets, we designed a species-specific probe for each target. For the pictures to your left, we test the EGF receptor, and to the picture of your right, we test PKM1. So the human-specific probes, in this case, is labeled in the green channel, only detect the EGF receptor in human-derived tumor. And the mouse-specific probes, in this case, is labeled in the red chromogen, only detect the low expression of EGF receptor in the mouse-derived stroma area. To the picture to your right, the PKM1, which is the endothelial marker, was majorly expressed by stroma cells. And you can see the very strong detection of the mouse-specific PKM1 probe, detecting the PKM1 in mouse stroma, but very low detection of the PKM1 in the human original tumor by human-specific probe PKM1. So using the 2.5 duplex assay, we can detect species-specific targets within the same regions of the patient-derived xenografts. In the slide 32 is another examples of the patient-derived xenografts using duplex assay to detect different markers. This time is the markers FGF receptor 1 and the TGF-beta 1. And you can see the human-specific probe, which is in green, only detecting targets in the human-derived tumor. And the mouse-derived probe, which is labeled in red, only detects signal in the mouse-derived stroma. So these probes are specifically designed to have no cross-reactivities between human and mouse. We name it as species-specific probes labeled as the no cross. For example, the TGF-beta probes for human labeled as human TGF-beta 1, no cross to mouse. So now we review some technical details of the duplex assay. We first validate whether this assay have the reproducibilities within the same test. So in this slide, you see the very high consistency between slides within the same run using the same oven. This is oven number 1. On the oven number 2, you can observe the similar very high reproducibility between the slide to slides. We also validate the reproducibilities from day to day. So this is the duplex assay run by 3 individual days and using the probe TBP in green and poly2A in red. You can see the very identical staining patterns from experiment done on different days. We also tested this assay using different operators. So on this example, you can see the operator number 3 and the operator number 4 running this assay on different instruments on different days, and you can see very similar results. All these results showing the 2.5 duplex assay have very high reproducibilities through day-to-day, run-to-run, and operator-to-operator.
Thank you, Ming-Xiao. So in conclusion, ACD continues with product innovation and now introducing the RNAscope 2.5 HD duplex assay. We've demonstrated robust results in different tissue types amongst different targets and applications as Ming-Xiao delved into the details. And we have thoroughly tested in-house, demonstrating reproducibility within and between runs, different ovens, and operators. I would also like to bring your attention to additional resources available to you during the course of running the RNAscope assay. Please use the support tab on the ACD website to access these. And lastly, we're always happy to assist you with any questions or concerns you may have with regards to your RNAscope assay. You may reach us through any of the methods listed in this slide. With this, we come to the conclusion of our presentation, and we will address some of the questions that you may have for us. We have a question here that says, what are the main differences between the old and the new kit, the 2.5 HD duplex assay and the old duplex assay? So the main difference between the two kits is that both the channels are now balanced. The green and the red are both amplified to the same level. Also, in the older duplex kit, there was an issue with the green signal fading, and that was a concern, which has now been corrected with the 2.5 HD duplex assay. Can the 2.0 C1 and C2 probes be used with the 2.5 kit? Yes, the C1 and C2 probes can be used with the 2.5 kits. There is no probe design change between the older and the newer kit. The C1 and C2 probes are still can be used with the newer 2.5 duplex kit. When will the duplex kit be commercially available, and what is the cost of this kit? The kit is currently in early access stage. If you are interested in trying this kit, please reach out to your regional account executives and they can assist you further in this matter. Are the green and the red the only available color options? Yes, the commercially available standard color options are red and green, as we have found the contrast works well for most markers. What other mounting mediums are suitable for the RNAscope 2.5 HD duplex assay? We have currently validated our assay with vector mount and found that it preserves the signal intensity of the chromogens well. We have not yet validated with any other mounting mediums and therefore recommend strictly using vector mount mounting medium for this assay. Can we run fresh frozen tissues with this kit? Our R&D team is in the process of extensively validating this kit for fresh frozen samples. Once we have validated this, we could provide some recommendations for appropriately permeabilizing this sample type for a successful RNAscope run. Can the signal from this kit be viewed under a fluorescent microscope? This kit is intended for use as a chromogenic kit, and the signal can be visualized using a regular light microscope. If you are interested in fluorescent detection, we recommend using the RNAscope fluorescent detection kit. Is this kit available for automated platforms? Yes. A duplex kit with red and brown combination is available on the Leica platform. A duplex automated kit for the Ventana platform is on our product roadmap. With those questions, we have now come to the conclusion of this webinar presentation. Thank you all for joining us today. We look forward to speaking with you via email. If we have not addressed your questions during this webinar today, we will get back to you on those offline. Thank you.